Hey guys, welcome to AP Biology, and we're going to talk about species concept 2. What is a species? So, we're going to talk about what a biological species concept is, which does represent or does emphasize reproductive isolation. We're going to look at some of those barriers that are both prezygotic and postzygotic in order to lead to that isolation. And we'll look at some of the limitations the biological species concept has, and also look at some other alternative sequences that we do see, or species concepts that we do see and use in biology, depending upon the situation. So, no, that is not a real organism. Eudyptes is the genus for the rockhopper penguin, but of course, that is a Photoshop thing. Alrighty, let's go ahead and look at our next slide. Excuse me. All right, so fossil record shows two basic patterns of speciation. One's called anagenesis and one is cladogenesis. Anagenesis is basically the small accumulation of changes that leads from one species into another with not really increasing biological diversity. Cladogenesis, or branching evolution, is going from one to more than two or more other species, and it does result in an increase in biological diversity. So think about that a little bit, and what form of speciation did you discover when you did the Anole lab, the Holy Anole lab? Okay, just think about that, and you should come up with an answer. Alrighty, as I said earlier, the biological species concept does emphasize reproductive isolation. So once a species is formed, then that species is not going to breed back with its parent species. And if you had finished the Anole lab, the example of Module 4 is a good example of how these reproductive isolating barriers can evolve, okay, and how they play out. So this first biological species concept first proposed in 1942 by a man named Ernst Mayr, and he basically says a species is a population or group of populations whose members have the potential to interbreed with each other in nature to produce viable, i.e., living, fertile offspring, but who cannot produce viable, fertile offspring with members of other species. So, that in nature is a really important concept because, yes, we can potentially take species, different species that are closely related from nature, bring them into a lab or in a forced breeding situation, and will be able to produce living, viable, reproductive, fertile offspring. But in nature, they would not normally ever meet. So just want to emphasize that one fact. Okay. The other thing is species are based on their ability to fertilize each other. Members of the same species can fertilize each other and again produce viable, fertile offspring. So here we look at 6 different humans, and there's a lot of phenotypic diversity there. And some people might say, "Oh, there's a bunch of different races there." In biology, race doesn't. There's no such thing as a race. We're species A or species B. And with biological species concept, all humans are members of the same species. We just show different phenotypic variations, slight variations, whether it's in skin color or eye shape or hair texture or blood type, things of that nature. Those are all superficial. They don't really mean anything. There could be local adaptations for certain environmental conditions or just a result of the segment of the gene pool that led to that beginning of that particular population. Again, thinking about the founder effect, because remember, modern humans came out of Africa, a relatively small population, not the whole gene pool. And then once they came out of Africa, they split up. One group went kind of east towards Asia, another group kind of went north towards Europe, and different environmental conditions. And, again, smaller portions of that gene pool led to the phenotypic differences that we do see in human populations today. So all members look very different, but all members of the same species. look really similar, but these are members of different species. These are eastern and western meadowlarks. Okay, they will not breed with each other. To you and I, they look exactly the same. They live in very similar habitats. However, they have different songs, and therefore the females do not recognize the males who are singing a different song. And they live in similar habitats, but that habitat is divided. The western meadowlarks are found west of the Rocky Mountains, and the eastern meadowlarks are found east of the Rocky Mountains. That's a The Rocky Mountains is a huge geographical barrier for the movement of species east and west. So when we talk about these barriers can either be pre-zygotic or post-zygotic. And again, what is the zygote? The zygote is a fertilized egg. Okay, so pre-zygotic barriers are ones that are going to occur and do not allow fertilization of the egg, and post-zygotic barriers are barriers that prevent the egg or the individual from producing viable fertile offspring, and it happens after fertilization of the egg. Okay, so that's the basis of that particular slide. So, what are the prezygotic barriers? Here are the 5 prezygotic barriers that you need to know: habitat isolation, temporal isolation, behavioral isolation, mechanical isolation, what I like to call it don't fit, okay, and gametic isolation. So, habitat isolation is when organisms do not occur in the same habitat. Um, so think about that with our anole examples. Okay, in our anole examples, we basically had 4 different habitat ecomorphs. Okay, and most likely the canopy anoles are not going to be interacting with the ground or grass species anoles. So that's an example of habitat isolation. Or the western meadowlarks are not going to interact with the eastern meadowlarks. They're isolated by the Rocky Mountains. Um, the temporal isolation. Temporal means time. Okay, that means they may be breeding at different times, as the picture below showing a wood frog and a leopard frog. The wood frog breeds a little bit earlier than the leopard frog does. Okay, and so they're going to be isolated temporally. But as I said on an earlier slide, multiple barriers generally exist. So let's look at these frogs. First of all, a wood frog and a leopard frog. Wood frogs are found more in drier habitats. They're found in woodlands. Leopard frogs are found in ponds. So there's habitat isolation. Wood frogs breed earlier than do leopard frogs. That's temporal isolation. Wood frogs and leopard frogs both have different types of mating calls that they don't recognize each other as members of the same species. So that is a form of behavioral isolation. So there's 3 right there. Okay. Another form of behavioral isolation would be like the birds of paradise. And we looked at the Parodia video clips and things where we saw all these elaborate dances and stuff. This is a bluebird of paradise. It hangs upside down and has a very atypical display for birds. It hanging upside down, shaking its feathers. The male has a very distinctive call that other females of the same species recognize, but other birds don't. Okay, these guys are more of a lower mountain species versus a higher altitude mountain species. So again, there's some habitat isolation going on there. Prezygotic barriers, again, habitat isolation. Here's a picture showing salamanders that are found from Northern California all the way down to Southern California. And what we show here is we see the San Joaquin Valley in the center part of California. That is a very dry and hot, low altitude valley. And as you can imagine, salamanders don't like those conditions. Where on the coastal range is very humid, lots of fog, lots of rain, beautiful habitat for salamanders. And then you have the windward side of the Sierra Nevada mountains. Okay, again, relatively damp and wet, great salamander habitat. So, as these salamanders, which originated in kind of around the Oregon Pacific Northwest area, as they expanded their range southwards, they avoided this drier habitat and the population became split. And as that population became split, they acquired different characteristics, different adaptations for their different, slightly different environments. So by the time you get down to the southern end down here, as you can see from these pictures, they look really different. One is spotted, one is not. They don't recognize each other as members of the same species. So that's an example of habitat isolation. We'll come back to this picture a little bit later. Mechanical isolation, i.e., it don't fit. Now, here are two dogs, and you can see they're vastly different in size. It would be very difficult for these two dogs to mate, even though they are members of the same species. All dogs, domestic dogs, are Canis familiaris. However, due to our artificial selection on these breeds, they cannot breed together. Yes, we could take some sperm sample out of probably the smaller dog and put it into an egg in the bigger dog, and we could potentially produce some weird mix coming out of that. But physically, they wouldn't be able to do it on their own. We also see mechanical isolation in flowers. Here we see bees able to get into the nectary and it's able to get pollen on it and stuff, but the larger bee can't get into there. So flowers also have mechanical isolation. And finally, gametic isolation. Okay, here the gametes, here we see plants. This is pollen. Different pollen grains have different proteins on them, and the pollen grains may not germinate on the female part of a different species of plant, but they will on members of their own species. Okay, if you open up this slideshow on your computer, you can clip on this clip and watch a 2-minute video. It's about coral spawning. And basically, when corals spawn, they release tremendous amounts of eggs and sperm into the water at the same time, usually around the full moon in August. And it's like swimming through a snow globe. You'd be surrounded by sperm and eggs. Sounds wonderful. But all different species. So how does one species of sperm find the right egg? Well, there's a couple of different ways. There's some chemical signals that help them to move, but also a sperm from let's say a sea cucumber will not be able to fertilize an egg from a coral. There's different proteins that basically prevent that from happening, almost like cell communication. So that won't be able to happen. And think about our anoles. What are all the different types of barriers, prezygotic barriers, existed with our anoles? Think about that a little bit. You should be able to come up with at least 3 prezygotic barriers that prevent anoles from mating with each other. Then we get to postzygotic barriers. These are barriers that occur after the fertilization of the egg. So here's fertilization of the egg right here. Number 1, reduced hybrid viability. What does that mean? That means that the hybrids die before they are able to reproduce. They either miscarry, you get fertilization, the embryo doesn't implant, the organism dies as a fetus or whatever, or they just die before they're able to breed again. Okay, so they don't live. Reduced hybrid fertility. This is when your hybrids are sterile. So you take a donkey, you mate it with a horse, and you get a mule. That's the only way you're going to get a mule, because mules are sterile. Horses and donkeys have different numbers of chromosomes, and it won't work. Okay, but you do get a nice strong pack animal called a mule out of that crossing. You just can't make mules from mules. You got to get a donkey and you got to get a horse and breed those. The last one might be a little bit harder to understand, hybrid breakdown. This is when you do produce viable, fertile offspring, but those offspring are going to not produce as many offspring themselves, and usually over a period of a few generations, they basically disappear. So here you have a species A on the left and species B on the right. When they cross-pollinate each other, they produce species C in the middle. Notice how many seed heads are on this wheat plant in species A and how many there are in species B over here. But Species C, the hybrid, has a few. It's not going to produce that many seeds, therefore, it probably is not going to make a big contribution to the gene pool, and they just basically disappear after a few generations. What are some alternative concepts of species? Well, let's think about this, okay? Sometimes they emphasize the processes that unite the members of a species, such as the ecological species concept. We define the species in terms of its ecological niche. Is it a producer, i.e., an autotroph like plants? Is it a consumer, i.e., heterotrophs that eat producers and other consumers? Or is it a detritivore? Detritivore is simply a decomposer. Okay, anything that's breaking down detritus, which is dead and dying material, leaves, plants, rotten logs, things of that nature. Now, the oldest species concept that we've ever used is basically, what does an organism look like? Okay, and again, it's still the most practical, and it defines a species by a unique set of structural features. Does it have wings? Well, if it has wings, does it have feathers? Okay, if it has those two, it's a bird. All right, so on and so on. Now, you can sometimes get in trouble with a morphological species concept, because sometimes species, due to similar environmental conditions, species from very different backgrounds can begin to resemble each other. That's, again, that process of convergent evolution. The other thing that is an example, remember, with the biological species concept, we have to make sure that they have the ability to interbreed and produce viable fertile offspring. So, what are some problems with that? Well, think about species that reproduce asexually. Can't quite use a biological species concept for that. So think things, especially things like bacteria. For bacteria, for the longest period of time, we used a morphological species concept. Are they round? Are they rod shaped? Or are they spiral shaped? Do they absorb a type of stain called a gram stain? And if they are, they're gram positive. If they don't, they're called gram negative bacteria, which is related to their cell walls. But that was about it. It wasn't until we started using DNA to classify the bacteria that all of a sudden bacterial diversity really started to come into light. Um, but also, what about species that are extinct? We can't define those species by biological species concept because we've never seen them breed. Think of things like dinosaurs. So with dinosaurs, we have to use a morphological species concept, and we have to look at their bones. And similar bones would, allowed us to classify those species as being similar or closely related. Nowadays, with the advent of cheap genomics and the ability to sequence entire genomes both cheaply and quickly, we can now look at nucleotide sequences, or we can look at protein amino acid sequences, and that leads to molecular homologies. The closer two DNA sequences are, then we assume the closer they had a last common ancestor. The more divergent those DNA sequences are, then we can say, well, that their last common ancestor was much further back in time. So with our cousin, the chimp, okay, we share about close to 99% of our DNA with a cousin. With each other, we share like 99.99% of our DNA with the person sitting next to you. Whether you're in Bombay or you're down in Papua New Guinea or in South America or in Finland, you share about 99.99% of your DNA with that person sitting right next to you. So, pretty interesting stuff. So, how does speciation happen? Two major ways we can talk about it: allopatric speciation and sympatric speciation. If you remember that word allo, we saw that with allosteric regulation of enzymes, and that meant allosteric meant other place. So there was a molecule that bounded not to the active site, but to another place on that enzyme. So allopatric speciation, if you have any Latin, patria means fatherland, like patriarchal societies, things of that nature. Allopatric speciation means that somehow that original population has been separated, and there's no gene flow between it. Sympatric speciation is when we see new species arise within the midst of the parent species. So one great example of allopatric speciation is the Grand Canyon. I'm not sure how many of you have ever been to the Grand Canyon, but it's a big old hole in the ground that's about a mile down. It's extremely wide from the north rim to the south rim, and it's very rugged terrain. Now, the Grand Canyon looks old, and in terms of human lifetimes, it's relatively old. It's between 1 and 2 million years old. I can't remember the exact date on the Grand Canyon, but in terms of evolution or geology, that's a blink of the eye. And the way the Grand Canyon formed was the Colorado Plateau was generally relatively flat and broad. And then about 1.5 million years ago or so, that Colorado Plateau began to lift up. It became uplifted. And as it was doing that, the Colorado River was up there and it just started cutting down through all these soft sandstones and things like that. And that created the Grand Canyon today. Well, when that happened, it split these populations of squirrels, one on the south rim and one on the north rim. Genetically, they're almost identical, but they have different variations. They look different from each other. If you take one of these north Kaibab squirrels and put it on the south Abert Rim side, the two squirrels don't recognize each other as members of the same species. We could make them breed and we could produce viable fertile offspring, but remember, the biological species concept says in nature. Okay. Ring species, we mentioned this with the salamanders, another piece of great evidence for how allopatric speciation occurred. Remember, this population became separated as they expanded southward, and by the time they come back together again on the southern end, they no longer recognize each other as members of the same species. Adaptive radiation. So this is basically what kind of happened a little bit with our anoles. Okay, we had our colonizer anole landed on an island or a group of anoles, and they underwent natural selection and adaptation in order to reduce competition. Because think about these anoles, they land on an island, there's no other lizards there, there's plenty of food, they go wild, their population grows really quickly. And what is the biggest competitor for any species? You got it, a member of its own species. So in order to reduce competition, maybe, and of course there's genetic variation, there's phenotypic variation in these guys. Some maybe have a little bit bigger toe tip pads, and so they're able to climb up to the top of the canopy and utilize the resources up there. Some have shorter legs, so they're able to go on the little twigs and get the food out there, and so on and so on. And eventually that leads to this diversity of anoles. Well, the same thing happened in Hawaii, except that the Hawaiian Islands have been around for a lot longer. Okay, Hawaiian Islands have been around for like close to 100 million years. And the finch species that was the founder is very similar to the purple or house finch that you might have at your bird feeders if you feed birds. And they underwent a very strong adaptive radiation to fill all the available niches that were available on the island of Hawaii. So we have nectar eaters, we have ones that are predators like little mini hawks and things. We have seed crackers, we have little insect gleaners, and lots of different niches that were available. And they underwent an adaptive radiation to fill those. Unfortunately, most of these Hawaiian species of finches are now extinct due to a variety of other things. This is a picture of the anole adaptive radiation. This is a phylogenetic tree in a circular form. Follow it through. This is where the original anole is. We see a split here, and this split led to this group over here, and the split in this direction led to all these other anoles. And again, this is all based on DNA evidence. All right, so just look at that, spend some time, stop, pause the video, and look at that so you see if you can understand. There's a little key down here showing you what's going on. Interesting graph to look at. And so plants do the same thing. Remember, Hawaii is one of the most isolated places on the planet. And for plants to arrive there, they had to have either floated there or arrived by wind or arrived by bird potentially. Okay, well, Hawaii has an incredible adaptive radiation of a group of plants that we call the silver swords. And the silver swords originally came from a small sunflower like plant found in California. It was only about 6 inches tall. And the silver swords have undergone an adaptive radiation into extremely tall plants and trees and things of that nature. I'm going to pause the video so I get going, then I'll finish up later. Okay, so getting back to the Hawaiian silver swords, again, as I said earlier, Hawaii is a very isolated place, and so in order for plants to get there, they have to travel either via animals like birds, or by the wind, or by floating. And as I said, the silver swords come from a plant called a California tarweed, which is a sunflower-like plant that does produce wind-dispersed seeds, and it made it over to Hawaii, and over millions of years, because remember the Hawaiian chain is 60 to 75 million years old, they evolved into a great diversity of species, from small shrub-like herbaceous plants, low plants like this green one up here in the corner, to trees that are 70, 80 feet tall, to these incredibly interesting silver swords that we do see on the slopes of the high volcanoes, to vines. They diversified very, very, very quickly. Um, Other things that have undergone adaptive radiations in Hawaii are the fruit flies, Drosophila. As I say up here, there are over 1,000 species of fruit flies on the Hawaiian Islands. There's not many more than like 1,500 worldwide. So Hawaii has this incredible diversity of fruit flies. And if you know fruit flies, you know those small little flies you see in the summertime. They basically feed on fruits and that are rotting and things of that nature. But in Hawaii, because there were so few insects there, they really diversified into pretty wide group of organisms. So they played the role of predators and some eating other smaller insects, herbivores, eating plants like the leaves and stuff versus going after the juices or the fruits and stuff. Nectar eaters, parasites, detritivores. Again, because there was a lack of competition, they underwent this adaptive radiation. In history, there have been many other adaptive radiations, and most of the adaptive radiations begin after a mass extinction event. I wish I had this picture as a little bit, went back a little further in time to the Triassic Permian boundary, because at the Triassic Permian boundary, there was a group of mammal like reptiles that were mostly wiped out during the Permian extinction. When they were wiped out, that allowed the small number of dinosaur-like reptiles that did survive to rapidly undergo a adaptive radiation, and they basically ruled the planet for 190 million years until 65 million years ago, when there was another mass extinction event that led to the loss of the dinosaurs and the adaptive radiation of the mammals. So we are currently in the age of mammals, because the mammals really underwent an adaptive radiation. I should say we're also in the age of birds. We have lots of birds. Now, you might think, well, where are all these mammals? Well, a lot of them are extinct, but most of the mammals that we have today, and we do have quite a diversity of mammals, are either bats or rats. So we have quite a few number of rodents and quite a few number of species of bats. So sympatric speciation is the opposite of allopatric speciation, is when a new species arises and there's no sort of geographical barrier between the two. Okay. In plants can actually undergo a speciation event in one generation. You're like, what? I'll show you how that can happen. It's generally a result in a non-disjunction event during meiosis. In animals, however, excuse me, oh, oh. In animals, sympatric speciation is often a result of a result of trying to reduce competition between organisms, like we saw with the anoles. So let's look at what can happen with plants. This is autopolyploidy. Let's break that term down. Let's go back there. Auto means self, poly means many, ploidy means sets of chromosomes. This is when a single species undergoes a non-disjunction event. So we start off with a parent plant that has 6 chromosomes. There's a non-disjunction event in during meiosis that produces a gamete that's unreduced. It is still diploid and it has 6 chromosomes. And then another plant, or even the same plant, if it's self-fertile, produces the other gamete, i.e., so one's going to be a pollen grain, one's going to be an egg cell that is also unreduced. And when they fertilize, you end up with a plant that is now, again, tetraploid, has 4 sets of chromosomes. This plant now can no longer breed back with its parent plant. Okay, it just can't. There's too many chromosomes. And again, like plants can do this. And you might be going, what's the chance of that happening? Well, just wait about another month or so and you go look out on your car, look at how much pollen is on your car, and then you'll see that the odds are that it's pretty likely that this can happen. Allopolyploidy, however, is a little bit different. Allopolyploidy, remember, allo means other, means that there are more than one species that are taking part in this particular process. Generally, the species have to be closely related. And what we see here is we have species A with 4 chromosomes and species B with 6 chromosomes. Species A has a meiotic error and produces a gamete cell that is also diploid with 4 chromosomes. Species B produces a normal gamete, and when the normal gamete fertilizes the abnormal gamete, you now end up with a hybrid that has an odd number of chromosomes. It's got 7 chromosomes. Generally, that hybrid is not going to be fertile. It can still produce gametes, and in this case, we see another meiotic error. There are no chromosomes lost, and so we have an unreduced gamete with 7 chromosomes that then gets fertilized by a normal gamete from plant species B, resulting now in a viable fertile hybrid that has 10 chromosomes. Okay, an equal number of chromosomes, and so on and so on. All right. And one of our best examples of this is our the wheat flour that you eat when you eat cookies and cakes and breads and things of that nature. Okay, where we had, again, this is something that you don't really need to know the detail. What you really need to take home is that plants can do this process where chromosome number changes due to meiotic errors, non-disjunction events during meiosis that leads to gametes that have unreduced number of chromosomes, and that through this process of random fertilization, we end up with these hybrids that now have a different number of chromosomes than the parent cell. And if these hybrids can now establish a breeding population, we now have a different species with a different number of chromosomes that can no longer breed back with the parent event. In autopolyploidy, this can happen in one generation. In allopolyploidy, generally it's going to take a couple of generations maybe for this to happen. Okay, so just some kind of interesting stuff. Sympatric speciation examples, already have the example of the anoles. African cichlids are a great example of this also with fish species. And again, this is in the Great Rift Lakes of Lake Victoria, Lake Kivu, Lake Tanganyika down here. I used to live right there where that arrow is, on the shores of Lake Kivu up in the mountains of the Nyungwe Forest. Beautiful part of the world. They call the Afro-Montane region or the Albertine Rift region. But anyways, Lake Victoria about 200,000 years ago completely dried up, according to geological evidence. There were no fish in there. When it started to fill back up again, fish got into there via little rivers and straight streams that feed into Lake Victoria and rapidly underwent an adaptive radiation. Currently today, there's well over 200 species of these cichlid fishes, although many of them are endangered due to habitat destruction, pollution, but mostly due to the introduction of invasive species that eat them. But anyways, these cichlid fishes, how do they make so many species in a relatively short period of time? Again, in order to reduce competition. Okay, it's not a purposeful thing. Some had some variations, and some preferred, the deeper water. Some preferred the shallower water. Some ate one food type. Others ate the other food type. Cichlids can be small little fish, about that big, to really big fish. And so they have a variety of food things that they do eat. And another thing about cichlids is that they have a very interesting jaw. They actually have two jaws. And if you remember what I said about gene duplication, when a gene duplicates itself, one of those genes is going to do the job that it's supposed to do. The other one is sort of free to potentially mutate and take on novel functions. Same thing with these cichlid fishes. The inner jaw, which you can't see in this picture, does the job of chewing and crushing and whatnot. The outer jaw now has a high degree of plasticity, meaning that they are relatively free to evolve. And these fish have some really bizarre niches. Like one only eats scales off the right side of other fish, another one eats scales off the left side of the fish. Some pluck the eyeballs out of fish, some chew the tails, some are algae eaters, some are snail eaters. Quite a great diversity, and many of these are very colorful, which is why hobby aquarists really like to have cichlid fishes in their tanks because they're really bright and colorful, and they are freshwater fish. Okay. Um, Other modes of speciation you might say we've already talked about convergent evolution with the ecomorphs, okay, in the plants, in the lizards, and also parallel evolution. Now, parallel evolution is probably these two terms can be a little confusing sometimes, and if you notice, look at they basically say the same thing. Similar environmental conditions lead to similar adaptations. Boom. Similar environmental conditions lead to similar adaptations. The difference is in convergent evolution, it says in different unrelated groups of organisms, meaning they have a distant last common ancestor. Great example, sharks and dolphins. Sharks, of course, are fish. Dolphins are mammals. Birds and bats. Birds are birds. Bats are mammals. Cactus and euphorbs. And you're like, what the heck are those things? Okay, well, you all know what a cactus is. Well, cactuses are only found, well, I should say they originally were only found in the New World, North, Central, and South America. Okay, and euphorbs are only found, or I should say, used to be found only in the Old World, Asia, Europe, and Africa. And if you go to desert systems in Africa, or parts of Asia, you will see plants that look just like a cactus plant. I mean, you would have a hard time telling the difference just by looking at them. Okay, they look exactly the same, but they are completely unrelated. They've just natural selection has landed on a kind of a series of phenotypes that works well in a desert system. And as we said in the movie, evolution does repeat itself. And that's what happened there. Parallel evolution, again, is similar adaptations in two related groups of organisms, i.e., they had a more recent last common ancestor. And this would probably be more of the case of the anoles. But a great example is marsupial and placental mammals. So remember, marsupial mammals are those things like kangaroos, things that have a pouch, and they are found pretty extensively, of course, in Australia, which doesn't have native placental mammals, and in South America. Now, South America has lost a lot of its marsupials because about 1.5 million years ago, Central American land bridge connected North and South America, and placental mammals went into South America, and they basically outcompeted the marsupials. Only one marsupial made it into North America, and you are all familiar with that. You see them dead on the road all the time. Those are possums. So let's look at some of these examples. We have the flying squirrel here that we have here in Alabama, okay? But in Australia, you have sugar gliders. They look exactly the same. They have the same ecological niche. One's a marsupial, one's a placental mammal. In Australia, we have crest-tailed marsupial mice, where we have our regular mice over here. You have marsupial moles over there, and here we have our regular moles. You have a thylacine or Tasmanian wolf. Unfortunately, these guys are extinct now. And of course, we have our wolf. There are wombats, which have a similar ecological role to our woodchucks or marmots. You have anteater-like marsupials compared to our South American anteaters and things like that. And you even have a thing that's almost like a cat. Fills the same niche as a cat does. But it's not a cat, it's a marsupial. It's a spotted tailed native cat or tiger cat. And again, it is not a placental mammal. It doesn't have a placenta. They'll be young or born very immature. They crawl up and go into a pouch. And the last slide is dealing with the rate of speciation. Darwin proposed a gradual, very slow accumulation of changes over time that led to new species. And we call that gradualism. Some people call it Darwinism, so on and so on. But back in 1972, Stephen Jay Gould and Niels Eldredge, who were studying the fossil record, particularly looking at snails, they didn't see a gradual change. What they saw were long periods of time with no morphological change in organisms, and all of a sudden they saw a completely new set of organisms that had replaced the original organisms. And I don't want to say all of a sudden, we're talking over about a 5 to 10 million year period, which is relatively quickly in looking at the big scale of evolutionary biology. But they basically said, well, the fossil record doesn't show gradualism. The fossil record shows these long periods of no change, followed by some sort of environmental disaster, and then very rapid diversification, and then again a long period of little change, and then rapid diversification, and so on and so on. So, which one's correct? They kind of both are correct, you know. And again, when you think about this, time periods, even if you have punctuated equilibrium, you are going to have these time periods where there's a massive change, but it still takes a pretty long time for those species to change within that massive changeover. So they're both kind of correct. I just want you to know what those two are. And that's it. I hope you learned something, and thank you for paying attention.